Hello everyone and welcome to my YouTube channel Perfect Lab. In today's video we will learn about the triple sugar ion test, a simple but important biochemical test used to identify bacteria based on their ability to ferment glucose, lactose and sucrose, as well as produce gas and hydrogen sulfide. We will also learn about the principles of the test, the composition of the medium, the procedure, and how to interpret the different reactions. So let's get started. Let's start our introduction by looking at what the TSI test is. So basically, the TSI test is mainly used in clinical and teaching microbiology laboratories to differentiate members of the enterobacterial side and other gram-negative bacteria based on their ability to ferment carbohydrate, produce gas, and produce hydrogen sulfide. TSI medium. The TSI medium is a solid agar medium, as shown here, prepared in a test tube with the agar solidified at an angle to form a slant and a deep bath. So this is the slant of the agar medium and this is the bath of it. The slant is exposed to oxygen, creating an aerobic environment, whilst the bath contains little or no oxygen to create an anaerobic environment. So let's see what goes on in the slant and in the bath in terms of bacteria metabolism. So the diagram here shows the slant portion of the TSI agar medium, which represents an aerobic environment because it is well exposed to oxygen. And bacteria growing on the slant use oxygen for aerobic metabolism. And in doing this, they first oxidize glucose, and once the limited glucose is depleted, many bacteria begin breaking down the peptones available to release ammonia that makes the medium alkaline. And this changes the phenol red, which is the pH indicator, in the medium to deep red or pink. And also some bacteria here may produce gas or hydrogen sulfide, and bacterial growth here is mainly observed along the surface of the slant where oxygen is readily available. The second picture here shows the bath of the TSI agar medium, which also represents an anaerobic environment because little or no oxygen is present here. And in this part of the medium, bacteria obtain energy by fermenting glucose, lactose, and or sucrose instead of using oxygen. And the sugar fermentation here produces acids that lower the pH to cause the phenol red indicator in the medium to turn yellow. And some bacteria here have also produce gas, resulting in cracks or lifting of the agar, whilst others produce hydrogen sulfide, which reacts with iron salts to form a black precipitate. And because the bath is anaerobic, bacterial growth occurs throughout the depth of the medium. Let us look at the medium composition of our TSI agar medium. So before we look at that, this is our TSI agar medium in our test tube, and this is the slant which supports aerobic metabolism and is exposed to oxygen, and this is the bath which also supports anaerobic metabolism and has little or no oxygen. And the final pH of this medium when prepared at 25 degrees Celsius is around 7.4 plus or minus 0.2. So the medium composition. So the first component of the medium is 3 types of sugars, and these are glucose and lactose and sucrose in the ratio of 0.1% to 1% to 1%. So what it means is that the glucose concentration to the lactose and sucrose is 0.1, 1, and 1. And these 3 sugars serve as a carbohydrate source that bacteria ferment to produce acid or gas. The second component is the pH indicator, which is phenol red, and this gives the medium the orange to red color at a neutral pH at 7.4 at 25 degrees Celsius. And this phenol red, which is the pH indicator here, turns yellow in the production of acid during carbohydrate fermentation and turns deep red when only peptone is catabolized, as this will produce an alkaline byproduct which will raise the pH of the medium to cause the phenol red to turn to deep red. The next component is sodium thiosulfate, and the sodium thiosulfate acts as the sulfur source, which provides sulfur for bacteria to produce hydrogen sulfide. And the ferrous sulfate here is acting as an indicator for the production of hydrogen sulfide. And what it does is that it reacts with the hydrogen sulfide produced to form a black ferrous sulfide, which is seen as a black precipitate to indicate hydrogen sulfide production. The sodium chloride over here helps to maintain osmotic balance necessary for bacterial growth. The peptone serves as sources of nutrient, especially nitrogen, amino acid, and proteins, which support bacterial growth. And this peptone can also be broken down by bacteria to produce alkaline substances or byproduct that will cause an increase in the pH medium to change the color of the phenol red to deep red. And another component is the meat extract and yeast extract that provide essential nutrients by supplying vitamins, minerals, amino acid, and other growth factors needed for bacterial growth and metabolism in the medium. So now let us look at the effect of pH on the pH indicator, which is phenol red in the medium. So at neutral pH, which is pH around 7.4 as shown here, the phenol red maintains an orange to red color. Now in an acidic pH, that is during the fermentation of sugars, the color of the phenol red changes from the red to orange to yellow as shown over here. And in alkaline medium, where the pH is above 7.4, and this normally happens when only peptone is catabolized to produce an alkaline byproduct that will cause an increase in the pH to change the phenol red from the orange to red color to a deep red color. Look at the bacterial metabolism in triple sugar ion medium. So first of all, the TSI medium differentiates bacteria based on their ability, 1, to ferment sugars, and 2, metabolize proteins, 3, produce gas, and 4, produce hydrogen sulfide. So let's start by looking at the carbohydrate composition in the medium. So 3 sugars are present in the TSI medium, and these are glucose, lactose, and sucrose, with glucose having a concentration of 0.1, lactose 1%, and sucrose 1%. So what it means is that the concentration of the glucose compared to lactose and sucrose is less, and the concentration of the lactose to sucrose is the same in the TSI medium. For aerobic and anaerobic metabolism, the slant, which is rich in oxygen, creates an aerobic environment, whereas the bath, which has little or no oxygen, creates an anaerobic environment. And these two conditions or environment allows bacterial metabolism to be observed under both conditions. So let's see what the color means in TSI test. So the yellow color over here indicates acid production from the fermentation of carbohydrate, and as this happens, the pH of the medium is lowered, that is, it falls below the 7.4, and as a result, the phenol red, which is the indicator in the medium, changes color from the orange to red to a yellow color as shown over here. Now, this color code also shows that there is no acid formation or no acid has been produced, and this also indicates that peptone is being metabolized to produce an alkaline byproduct such as ammonia that will cause an increase in the pH of the medium. And once this happens, the phenol red, which is the pH indicator, will change color from the orange to red to a deep or a bright red color as shown over here. And when there is no significant change in pH, the color of the medium remains unchanged, that is red orange as shown over here. Now let's look at how we can interpret the results. So we'll start by looking at when lactose and or sucrose fermentation occurs. So when this occurs, there's large amount of acid produced, and as this happens, the pH medium is being lowered to cause both the slant and the bath to change from the red to yellow color to a complete yellow color as shown here. When only glucose is fermented, the bath remains yellow due to glucose fermentation, but what happens is that the color of the slant changes back to red because glucose over here is or has rapidly been depleted, allowing the bacteria to begin metabolizing peptones aerobically. So when no sugar is fermented, no acid is produced, and as a result, only peptone is metabolized. So as this occurs, an alkaline byproduct will be produced, and this will cause both the slant and the bath to change from the orange to red color or red to yellow color to a deep or a bright red color as shown over here. So let's see other observations that are likely to be seen in the TSI medium test. So whenever there's a gas production, the gas produced during sugar fermentation is indicated by cracks, bubbles, or lifting of the agar as shown here. For hydrogen sulfide production, so what happens is that the hydrogen sulfide reacts with the ferrous sulfate in the medium to form a black precipitate, which is called ferrous sulfide, and this is usually seen in the bath as a black precipitate over here. So for the color key, whenever we see a yellow color key, it means that lactose and or sucrose fermentation has occurred, and as a result leads to the production of acid. So this color or this deep or bright red color indicates that olipeptone has been metabolized, and when the color of the medium remains unchanged, and this is the red to yellow color, so we see like no significant change in pH has occurred. So that means that like there is no metabolism that has occurred in the medium, and this black color indicates the production of hydrogen sulfide. Let's see what happens when olipeptone is metabolized. And one key information that we have to note is that peptone metabolism occurs mainly on the slant. This is because the slant is well exposed to oxygen to support bacterial aerobic metabolisms of peptone. And what you also have to note is that whenever peptone is being metabolized by these bacteria in the aerobic environment, an alkaline byproduct such as ammonia is produced. And this ammonia produced increases the pH of the medium. And once the pH of the medium is increased, the phenol red, which is the pH indicator in the medium, changes from red-orange to a deep or a bright red color as shown over here. However, in the bath, if there is no carbohydrate fermentation, the red-orange color of the medium remains unchanged. But in a case where glucose is being fermented, the red-orange color of the medium changes to yellow as shown over here. So in summary, Whenever peptone is metabolized in the TSI agar medium, the red to orange color of the medium changes to deep or bright red on the slant. And if there is no carbohydrate fermentation, the red to orange color of the medium remains unchanged at the bath, but changes color to yellow whenever glucose is fermented. Let's see the steps involved in performing the triple sugar ion test. The first step is to label the TSI agar tube with a simple identification, date, and any other necessary information as shown here. The second step is to flame to sterilize the straight inoculating needle until it is red hot and allowing it to cool for 5 to 15 seconds before using. The third step, using aseptic techniques, we pick a well-isolated colony from a fresh bacterial culture, and this is a bacterial cell grown in the lab within 18 to 24 hours. And then stop the bath by inserting the inoculating needle straight down through the center of the agar until it reaches near the bottom, and then withdrawing the needle along the same path to streak the slant by gently streaking the surface of the slant in a zigzag or a fishtail pattern. The slant inoculation helps to inoculate the aerobic slant, whilst the stab inoculation helps to inoculate the anaerobic bath. So once we are done with this, we then cap the screw tube slightly to allow exchange of gas during incubation. We then incubate the inoculated medium at 37 to 35 degrees Celsius for 18 to 24 hours in an upright position as shown here. And finally, we then examine the results. So let's see how we can interpret this result. So a yellow slant and a yellow bath mean there is a glucose and lactose and or sucrose fermentation. And what happens here is that during this fermentation, acid is produced that lowers the pH of the medium and causes the phenol red to change color to yellow as shown over here. And we normally report the result as this. So this is A, which indicates the production of acid in the medium, and which is often indicated by a yellow medium over here. For a red slant and a yellow bath, it means only glucose has been fermented, and the result is written as this, as K/A. So where the K shows an alkaline production, and which is indicated by the deep or the bright red color over here. For a red slant and a red or unchanged bath means no carbohydrate fermentation has occurred, and in this case, peptone has been metabolized. For the appearance of a black precipitate, usually in the bath, means hydrogen sulfide production. And the formation of cracks, bubbles, or lifting in the medium indicates the production of gas during the fermentation of carbohydrate. So for the color code, a yellow color code shows the production of acid as a result of carbohydrate fermentation, and which is normally indicated with alphabet A as shown over here. And for alkaline production, which is as a result of peptone metabolism, is indicated with a deep or a bright red color over here, and this is also represented with K over here. So whenever there is no microbial metabolism in the medium, it is recorded as NC, which means no change. Let's look at some important notes or tips in performing the TSI test. 1. Use a straight inoculating needle and not an inoculating loop. This is because a straight inoculating needle allows you to stab the agar in a single line through the center. Allow the inoculating needle to cool after flaming before picking a bacterium colony. This is because cooling prevents killing the bacteria and ensures a viable inoculum. 3. Use a pure and a fresh culture. And this is a bacterial cells grown in the lab within 18 to 24 hours. And what you have to note is that using an old or contaminated culture can give a false or unclear result. 4. Stab only once through the center of the agar as shown over here. This is because a single straight stab through the center ensures proper results. And 5. Read the result after 18 to 24 hours. And delayed reading after 24 to 48 hours may lead to incorrect interpretation because prolonged peptone metabolism can alter the slant reaction. Let's look at some interpretation again of result or reactions in TSI medium. So before we look at the interpretation of this result, I want us to know that in writing a result for TSI, we always write the bat result against the slant. So what it means is that the result over here is the result for the slant, and the one over here is a result for the bat. And one key thing that we also have to note is that the A over here means an acidic medium and it is indicated with a yellow color. The K means an alkaline medium, which is represented with a red color, and NC means no change. So let us look at the first result over here. So a yellow slant and a yellow bat means there is a glucose and lactose and or sucrose fermentation. For a red slant and a yellow bat means only glucose has been fermented. And for a red slant and a red or unchanged bat, it means no carbohydrate fermentation has occurred, but only peptone has been metabolized. And a red slant with a black precipitate, usually in the bat, shows hydrogen sulfide production. And cracks in the agar and the formation of bubbles or lifted agar in the test tube medium shows the production of gas during fermentation. So I would like to leave you with this triple sugar ion test result over here for you to go through for more and better understanding. So thanks for watching, and if you found this video helpful, please like, share and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.